Well, ladies and gentlemen, is this working, I guess? We're going to start the second day of the symposium with I think, a very timely topic. Whose tissue is it? Patient as product. Now, those of us who have been following this particular area with some interest over the years have realized that some very interesting things have been happening. That gene sequences for products of unknown function have been patented, not only by commercial concerns, but by the United States government as well. The commercial value of such sequences is undeniable, as the balance sheet for the first company that puts out a test kit for common mutations related to cancer is going to be able to show. We're also aware that genetic information is incredibly sensitive. It has the potential to change the person's view of their own personal future. It impacts their families and also their financial security. Does genetic risk for cancer or other conditions impart impact on the ability to get health insurance or employment, if it should be known, for example? What about diseases that have a strong social burden or stigma associated with it? HIV or AIDS or some other STDs. That information would also be contained in the blocks in the form of viral sequences or sequences of other pathogens. So against this background, I think our topic today is both timely and important. Our first speaker, Dr. Ellen Wright Clayton, represents a double threat. She is both a MD and a JD, and is on the faculty of the Vanderbilt University Schools of Medicine and Law. She is uniquely qualified to discuss our topic today. She chaired the working group which was convened by the National Center for the Human Genome Research and the Centers for Disease Control and Prevention to address the use of stored samples for genetic research. Dr. Clayton.
Well, it's a great pleasure and honor to be here today. And I am looking forward to what I understand will be a vigorous discussion. In recent years, problems posed by the use of human tissue samples for research have been discussed around the world, and more particularly, issues about the use of samples for genetic research has been the subject of even greater attention. A fundamental tension underlies all these discussions about the use of stored tissue for research. On the one hand is the desire to promote research to improve our understanding and to ultimately to promote human health. On the other is the recognition that research can pose risk to human subjects and that the people whose tissues are used about research may have concerns about those uses. The debate is further complicated by the fact that most of these risks are primarily psychosocial, which until recently have received little attention in our discourse, and by the largely unspoken fear that at least in 1997, increased knowledge may in fact not help people, particularly in the short run. And then another complicating factor of which I'm particularly aware is the concern that doing research without consent may pose risks or may undermine people's faith in healthcare institutions, a faith that I think is already actually quite tenuous in these days of mandatory moves to manage care and other problems of that nature. Now, I think that research within the military poses additional concerns. First of all, I think it's abundantly clear that the military has concerns about the health of its personnel that often or that can be taken to supersede their rights to give informed consent to treatment. For instance, relying in part on this risks, a court recently held that the Department of Defense could appropriately use investigational drugs in personnel who are heading into situations of combat without their informed consent. Similarly, relying on the interest of the military in being able to identify the remains of military personnel who are killed in action, courts have upheld the military's right to obtain tissue samples to form a DNA repository. a practice that was challenged by two servicemen on the ground that they feared discrimination if these tissue samples were in fact used for research without their consent. Now, in both of these cases that occurred in the 1990s, it is important to recognize that both courts went out of their way to say that what was involved was not research. And implicit in the court's analysis was the notion that had research in fact been involved, informed consent would have been required. So the goals of my talk today are to address some of the concerns about use of stored tissue samples, focusing particularly on concerns within the military, and to ask questions about what rules govern and ought to govern these practices. The first thing to say is, where are tissue samples? And the answer very clearly are just about everywhere. They exist not only in the DNA repository to which I've already alluded, but also to pathology labs and tissue repositories that are collected for other diseases. It's also important to recognize that tissue samples, where they exist, were collected for a variety of purposes. One, of course, is specifically for placement in the repository. Another is that tissue samples are, the overwhelming majority of tissue samples are collected in part for clinical care. Somebody has surgery, someone has blood drawn, and the tissue is saved for a period of time. And its explicit purpose was for patient care. And then another setting in which these samples are collected are, in fact, for the purposes of research. But one of the concerns that's raised is that the research that may be contemplated using the samples is different from that to which the patient or subject consented. Now, as I have thought about this subject over the years, it has become clear to me that the main issue is who are the potential decision makers and what role ought they to play. Traditionally, the way tissue samples have been used in research is that the person who holds the samples could largely do whatever he or she wanted with them. Their use of tissue samples was perhaps subject to concerns of the funding agency or perhaps subject to concerns of the entity that held the samples, but largely it was unconstrained. It seems to me, however, that there are a number of possibilities for people who could have, or people or institutions that could have a voice in how tissue samples are used for research. And these include not only the researcher and the researcher's institution, but also the IRB, the funding entity, and of course the patient, him or herself. And the question that I want to talk about today is the question of whether, in particular, the individual ought to have some role in deciding whether tissue samples that were obtained from them were used for research. Now, in terms of talking about the legal rules that govern this, the main focus of my talk today is going to be the rules that are codified in the federal regulations. Now, I have to say that the slides that I have are from the rules that apply to DHHS. As you know, these rules were extended to the DoD in 1991 under the so-called Common Rule, and in fact, the regulations applicable to DoD are essentially the same and are codified in 32 CFR. But my slides apply to these others. I will come back toward the end of my talk and look at Title 10, Section 980, and look at the impact of that on the discourse that is embodied in the federal regulations. Now, the first step in deciding whether what is required about research using human subjects is to recognize that under the federal regulations, certain kinds of research are exempted from the coverage of the regulations. Where the exemptions apply, neither review by an IRB nor informed consent are required. And the governing, all of this is in your handout, I am absolutely positive. But I want to point out a couple of things. The regulations exempt from IRB review research involving the collection or study of existing data documents, records, pathological specimens, or diagnostic specimens, if the information is recorded by the investigator in such a manner that subjects cannot be identified directly or through identifiers linked to the subject. Now, there are two important caveats that arise from consideration of this particular exemption. The first is on the issue of existing, namely, that if one is going to collect samples that one knows are going to be used for research in the future, the exemption doesn't apply. It doesn't matter how thoroughly anonymous they are. It doesn't matter whether they're identifiers or not. It simply doesn't matter. The exemption doesn't apply. The second question, and is one I know that Dr. Korn and I will engage in later today, has to do with the issue about identification. Read literally, what this says is that that in order to be exempt, it should be impossible to link the sample with the individual or the medical records with the individual. And I think the important language here to point to is the directly or indirectly. And so particularly the practice where researchers are given samples and or medical records, and someone somewhere retains a linkage or a key so that it's possible to go back and either get further information or go back and actually talk with the patient, I think falls outside the exemption. So, but it is important to recognize that at least some kinds of protocols are currently exempt under the regulation. In the document that you have in your handout, I think that there is growing consensus that even for exempt protocols, that there may be some utility for review at least by an IRB or at least for some sort of funding entity to look and see whether in fact the research is appropriate, whether and whether there are sufficient protections in place. So I would just say that at least right now that that protocols using anonymous samples ought to be reviewed by IRBs. Now, for non-exempt protocols, they have to be reviewed by IRBs to look at a number of issues to ensure that the risks to subjects are minimized and are appropriate in relation to the benefits that can be expected from the research, that the selection of subjects is equitable, that appropriate informed consent is obtained, and that adequate provisions are made to protect privacy and confidentiality. However, it is important to recognize, I'm going to go back to this slide in a minute, to recognize that full informed consent is not required under the federal regulations in all circumstances. And in particular, that institutional review boards can waive or actually eliminate or limit informed consent requirements under the following circumstances. If the research involves no more than minimal risk to the subject, the waiver or alteration will not adversely affect the rights or welfare of the subject, the research could not practicably be carried out without the waiver or alteration, and whenever appropriate, the subjects will be provided with additional pertinent information. Now, I know that you all had an extended conversation on the issue of minimal risk yesterday, and I'm not going to go into it further except to say that this is obviously a very vexed issue. The other issue that I think is very important in the context of research using tissues is the issue of practicability. And we have remarkably little guidance in this area about what constitutes impracticability, except I think it seems fairly clear to say that merely arguing that it would cost more to go back and talk to patients probably is not enough. Now, I have to say that there are points in which issues of cost become compelling, but I find it particularly hard to believe that where you have tissue samples from an identifiable human being that is still in your patient care system, and you have, say, 10 subjects, 10 patients whose tissue you're using, I find it hard to believe that it is impracticable to go back and talk to those people. So I think that even though going back and talking to those people does, in fact, add additional cost. The final point that I would make here is that increasingly, and I think this is a recognition particularly pertinent in 1997, that going back and giving individuals specific research results after their samples were used in research without consent is almost surely inappropriate. First of all, it's probably illegal under CLIA, since most research labs are not CLIA certified. But the other thing to be said is that at least in 1997, we know that research information can often be quite harmful to your health, because it puts you at jeopardy of losing your health insurance, other source of insurance, and your employment. So so I would say that probably it is almost never appropriate to go back and contact patients with information unless they agreed before participation in research that these results were information that they in fact wanted. Now, there are some important questions, I think, that remain. One is that I know that Dr. Korn is going to argue that if the interpretation that I have set forward, namely that we probably ought to be reviewing all protocols that use tissue samples, and that probably informed consent is almost always required when identified samples are used, that research is going to come to a grinding halt. One of the first questions is whether the interpretation that I have given for you is correct, and whether, in fact, the results are desirable. Now, first of all, I would say that the Office for Protection from Research Risks essentially has concurred with this interpretation. But the other point that I would make here is that other people have disagreed with these prophecies about what will happen to the course of research. And still others are engaged in devising strategies to enable patients to provide adequate consent for these uses. 2 weeks ago, I was at a meeting sponsored by the National Action Plan for Breast Cancer and also Priminar. I know many of you know this group, where we have made substantial efforts to allow patients at the time they're undergoing surgery for breast or other cancer to direct that their samples be used for research, and that they understand that they will not be getting results back, and they can place some limitations on how their samples are used. These protocols are already, in fact, in use in some NCI protocols, and we already have some data about patients' general willingness to participate in this kind of system. So I have to say that as a practical matter, one major concern is just whether patients will permit this, and I think the answer, at least in many populations, is that they almost surely will. These efforts to obtain patients' consent are driven not only by concerns about the need to comply with the law, but also by the recognition that IRBs, under the current regulations, are prohibited from considering certain kinds of consequences in research. In particular, they, and I know Bill Freeman and I don't completely agree about this either, but at least under my interpretation of the regulations, IRBs are prohibited from looking at the future use of research results in deciding whether protocols can go forward. Now, some of us would argue that this means that IRBs cannot look at the issue of the social harms that may follow from research. Now, we certainly know that in the past there have been some research protocols that look to create more social harm than any benefit, and that have been stopped. The most potent example of this is the XYY newborn screening program that was proposed in Boston and that was ultimately killed because of the concern that the children who were detected to have XYY would suffer tremendous harm under that protocol. Now, unless we are to conclude that these kinds of social harms are absolutely irrelevant, it seems that someone ought to be able to consider these. And if IRBs cannot, then it seems to me that some other person or institution ought to be able to consider these potential harms. And one of the possible people who could make this kind of argument or have this kind of consideration is the patient, him or herself. I think another issue that remains is whether individuals ought to have any voice in deciding whether their truly anonymous samples are used in research at all. The final question that I want to turn to, however, is the impact of this statute, 10 U.S.C. Section 980, which I know came up in conversation yesterday. And the question that I want to address this to is a question that we looked at actually earlier in the talk, namely whether there are compelling military reasons that justify research without consent that do not exist in the context of the civilian population. In other words, whether the same kinds of arguments about using pyridostigmine and botulinum vaccine or creating the DNA repository exist in the context of research with the military, or whether, in fact, this statute forbids that kind of analysis. And in order to add, and as one reads this statute, it seems pretty clear on its face that this statute is in fact more restrictive than the federal regulations, even those embodied in Section 32 of Title 32 of CFR, and would suggest that informed consent has to be obtained in more situations even than are contemplated under the federal regulations. So in order to figure out what this particular statute was all about, I went and looked at the legislative history. Now, as a matter of statutory interpretation, if the language is clear on its face, you're not supposed to go look at the legislative history, but I went and looked at it anyway because I thought it would be helpful. And I think it's worth noting that this provision was initially put in place in 1972, or something relatively like it. And the context is interesting. I mean, first of all, this was at the time of the initial revelation of the Tuskegee experiments. I guess everything that goes around comes around, of the Cincinnati radiation experiments, which were funded by the DOD, and by Dr. Beecher's article that appeared in the New England Journal of Medicine. And interestingly, this particular provision, the notion that one could not do research using federal funds without informed consent, was attached to a DOD appropriations bill. That's where it initially came into the law. The exact language was, none of the funds appropriated in this act shall be available for any research involving uninformed or non-voluntary human beings as experimental subjects. Now, at the time, a couple things interesting. It was initially proposed that this provision, that informed consent was always required, apply to all federally funded research. That was rejected by the House on the grounds, first of all, that this was a DOD bill after all, and that the House felt that they had not adequately considered the issues about whether informed consent was required. It's important to recognize that this was put in the Department of Defense appropriation bill before the passage of the National Research Act, before the commission that promoted the Belmont report, and before the adoption of the federal regulations that I've just discussed. This became part of Department of Defense a long time before that all happened. The other thing to be said here is that that language was rejected at that time that said that these monies could only be used by the Department of Defense for research that was particularly necessary for the DOD. In other words, to involve military subjects in research, that the research had to be specifically related to the military. So that was rejected. The other thing I think that's important to recognize here is that the legislative discussion that went around the adoption of this language focused on whether it would prohibit allowing the military to participate in testing new airplanes, testing military equipment. And the concern that was raised about this bill was not about whether military personnel could participate in biomedical research, but whether, in fact, they could participate in other things that were peculiar to the military. So this particular language that I just talked to you about continued essentially unchanged in every subsequent appropriations bill until 1991. at which time additional language was added that said that you couldn't use uninformed or non-voluntary subjects except in the setting where there was where the research could be beneficial to the subject, in which case proxy consent was required. And it was not until I'm trying to think of it. It was not until actually 1983 that this language that's up here actually was taken from the appropriations bill and actually put in the statute books. It is a change from the language that had been adopted in the previous year, and it actually, no reasons were given for its being put into federal law or for the change in the language, except that Senator Tower felt like this was merely a technical amendment. Now, what can be said about the impact of this particular statute? One is that it does predate the federal regulations, and so it's pretty clear that the understanding of the issues of informed consent and research predated that of the considerations of the Belmont report. The other thing I think that can be said about this is that it seems fairly clear to me that Congress intended that research in the military at least be not more expansive than that applied to civilian populations. I think you could read the language and say, in fact, they intended it to be more restrictive than that available in civilian populations. But at least it would be hard to say under this particular statute that research is more permissible than that in civilian populations. So to summarize quickly, the position that I am articulating here is that the use of stored tissue samples for research does raise substantial questions, that under the federal regulations, at least IRB review is required, and the possibility of informed consent needs to be considered wherever identified or identifiable samples are used for research protocols, and that the conditions for permitting IRBs to waive or limit consent are in fact relatively narrow. And so what I would say is that it seems to me that under federal law, that informed consent of the patient is at least very often required. Having said that, however, I strongly believe that it is not only legally required, but also quite possible to accomplish this, and that as we look to looking at retrospective use of samples that were obtained in the past, that we have to take a realistic look about our evolving understanding of the mandates of informed consent and come up with solutions that allow us to use samples into the future. Thank you very much.
Our next speaker is Dr. David Korn. He comes to us from Stanford, and in that institution has served in a variety of capacities: Chairman, Department of Pathology, Chairman, University Committee on Research, University Hospital Board of Directors, Lucille Salter Packard Children's Hospital Board of Directors. He's also been the Dean of the Medical School, the Vice President of the University. He's been the past President of the American Society for Investigative Pathology, formerly known as the American Association of Pathologists, a member of the Board of Directors and Executive Committee for the Federation of American Societies for Experimental Biology, FASEB, member of the Board of Directors Association of Academic Health Centers, a founder of and member of the Board of Directors and Executive Committee member of the California Transplant Donor Network, member of the IOM and the National Academy of Sciences, and by presidential appointment, the chairman from 84 to 91 of the National Cancer Advisory Board. He's been intimately involved in clinical research and practice, but he's also been a key player in the process of scientific peer review as a member of the editorial boards of several influential journals, including the Journal of Pathology, the Journal of Biological Chemistry, and Human Pathology. His primary research interests have included molecular biology of human DNA replication, and he is currently writing and speaking about health issues and science policy. The reason that he may be here with us today is that he's already in D.C. on sabbatical from Stanford. He came here voluntarily as a distinguished scholar in residence at the American Association of Medical Colleges and as a senior scholar in the Health Science Policy Administration at the Association of Academic Health Centers. Dr. Cole.
Good morning. As some of you may surmise, after being dean of a medical school for 11 years, I came east for R&R. So it was voluntary. I want to talk about genetic privacy and the use of human tissues in research and present a somewhat different perspective than Dr. Clayton did, although I think there are some areas where we probably agree, a few at least. And let me begin this way. It's quite clear that dramatic advances in science and technology that have been significantly stimulated by the Human Genome Project have already led to remarkable progress in molecular genetics and our understanding of normal and abnormal genomic structures. These advancements have already transformed biomedical research and promise equally profoundly in coming decades to transform the practice of medicine as well as strategies of health promotion and disease prevention. But at the same time, these accomplishments have generated powerful societal concerns about the acquisition, protection, and use of information that's considered to be intensely personal and private and may sometimes be predictive. These are very complex issues. They're very passionate. They arise from strongly held beliefs about individual autonomy and privacy, and they lie at the intersection of patient diagnosis and management, biomedical and other research, and public health. And in recent years, these concerns have come to very sharp focus on the nature of the informed consent process that should guide genetic testing and the use of human tissues in research. In recent years, these issues have received considerable attention in two different venues, one professional, the other legislative. The former has been reflected in position papers and other pronouncements from a variety of working groups and committees, many sponsored by the Ethical, Legal, and Social Implications Program, the ELSI program of the National Human Genome Research Institute, from genetic societies and from patient advocacy groups, particularly, as Dr. Clayton referenced, the National Action Plan for Breast Cancer. In my view, the contributions from these groups not surprisingly reflect their composition and offer a heavy dose of bioethical, legal, and patient advocacy perspectives, but a troubling deficiency of input from the broader scientific community, and especially from scientists who are increasingly using human tissue samples to gain powerful new insights into diseases that lay a heavy burden of morbidity and of human and financial costs on our society. The other venue, the legislative venue, is represented by sundry bills that have been introduced into the Congress and state legislatures and by laws recently passed, enacted in many states. Although a few of the congressional bills deal more broadly with privacy by addressing the confidentiality of medical information and the medical record, most of the efforts have focused more narrowly on genetic privacy and the discriminatory misuse of genetic information in employment and in the insurance markets. Unfortunately, many of these initiatives, and especially within the states, appear to be hasty and inadequately considered responses to public pressure, and most, if not all, bear the scars of heavy lobbying by the insurance industry. As a consequence, they tend to be confounded by semantic ambiguities that make them inadvertently overreaching, while at the same time they're compromised by exceptions on behalf of special interests deemed to be of overriding public or private interest. What I shall attempt to do this morning is to provide some context to these discussions, make a few general observations about the deliberative processes to date, and then offer a set of specific recommendations that I hope will further the discourse and maybe prove fodder for a debate later on this morning. First of all, it's important to recognize that the history of medical progress is deeply rooted in the careful study of normal and abnormal human structure and function, and that our nation's hospitals, and especially the academic medical centers, collectively contain an enormous archive of human tissue samples comprising a unique resource that reflects the prevalence and protein expressions of human disease over time. These specimens were removed for medical reasons under sparing consent language that usually included a proviso for research and educational uses, were submitted to the pathology lab for routine diagnostic evaluation, and then portions were permanently stored as part of the patient's medical record in accordance with sound medical practice and accreditation and legal requirements. Although not collected for research purposes, these specimens have served as a rich source of materials for clinical pathologic investigations that have provided, over the past more than 100 years, most of the vocabulary and much of the foundation of modern medicine. Yet the results of the studies have historically been primarily of public benefit and of little immediate consequence to individual patients or sources from whom the tissue had been obtained. And accordingly, as Dr. Clayton has said, the practice and standard of informed consent for this vast body of research has been minimal, and let me say right now, inadequate. What has changed now, of course, has been the introduction into practice of powerful new methodologies by which the pathologist and other investigators can use fixed, paraffin-embedded, even sectioned tissue specimens to demonstrate specific abnormalities of gene structure and expression, most of which turn out in the course of events to be of somatic origin, but some of which seem to be present or may be present in germline DNA and therefore heritable. The power of these approaches provides unique insights into the mechanisms of human diseases that cannot be obtained by other means, and they offer the promise of major advancements in diagnosis, prognosis, therapy, and even prevention. Yet by their nature, the results may on occasion be construed to have major predictive consequences for individual patients and their families. This fact has wrested the entire topic of research on human tissue samples from its historic state of repose and obscurity and thrust it sharply into the consciousness of a public already uneasy about erosion of privacy, fearful of discriminatory misuse of private information, and very susceptible of being stirred by anything that contains the iconic words gene or genetic. Now, the deliberations conducted to date by these various distinguished working groups and committees, in my view, have been disappointing in four major respects. First, they've contributed to semantic ambiguity by failing to distinguish between two different issues. One, genetic testing, which I argue should be defined narrowly and which raises legitimate considerations of definition and appropriate informed consent, and genetic information, which can be obtained or inferred from a myriad of clinical and research sources, and which raises different concerns of protection of privacy and confidentiality. Even more remarkably, with the single exception of one committee, none of the published proposals has taken note of, let alone distinguished between, somatic and germline mutations, their entirely different predictive implications. Second, it's been too readily accepted that genetic information is unique and different in kind from all other private, sensitive, and even predictive information that may exist in a medical record. To the contrary, I would argue that the difference is not so much qualitative as one of degree, and this becomes very clear when one attempts to separate genetic information from other medical information that may be present in a clinical or research record for purposes of restriction, protection, or regulation. The problematic concept of uniqueness is also important for another reason. It provides much of the conceptual underpinning for the proposition that patients or sources have enduring property rights in their excised tissue samples. And this, in turn, undergirds the elaborate and stringent informed consent procedures that have been proposed for this research. Third, it is disappointing that in attempting to deal with issues that largely center on preventing the misuse of genetic information developed in research, so much of the effort to date, all of the effort to date, has been expended not in trying to strengthen the protection of that information, but rather in burdening the conduct of genetic inquiry and erecting new barriers to the ongoing creation of knowledge. And finally, as noted above, the proposals reflect a heavy bioethical and legal perspective, but are rather light on contemporary medical and biomedical research perspectives and insensitive to scientific opportunity. In my view, these proposals come down too heavily on the side of private interest at the expense of public benefit, and thereby distort the delicate equipoise that must always be respected in research involving human subjects. Now, let me offer some recommendations and observations for consideration. Number one, much more careful attention must be paid to the definition of terms to enhance public understanding, sharpen the focus and precision of public discourse, and avoid both confusion and unintended consequences. In the context of contemporary molecular biology, the terms genetic research or human genetic research are exceedingly broad. The latter, for example, would encompass any research study, irrespective of purpose, that uses experimental systems derived from human cells or tissues and examines any nucleic acids or gene products, and that doesn't leave much outside the pale. Similar difficulties arise with terms like genetic sample, any specimen that contains DNA, or genetic test, any test applied to any specimen that can reveal genetic information. These terms, as commonly used, are far too inclusive and far too imprecise for using them without careful modification as the basis for new research guidelines or regulations. Two, genetic testing, appropriately defined, should unarguably meet a high standard of informed consent. However, the definition of a genetic test should be narrowed to focus on the purpose of the study rather than on the research methodologies that are used. For example, such a test might be one that is carried out on individuals or populations to determine the presence or distribution of particular heritable risk factors of established predictive significance for purposes of genetic counseling or purposes of medical management, or for reasons of public health. Research studies on human tissues removed for medical reasons ordinarily should not be defined as genetic tests, even if they involve examination of genetic structures and gene function. Research results are nascent data in a sense that they cannot be fully interpreted until they've been adequately replicated and validated. Only then can the medical community determine whether the results provide the basis for a useful genetic test that should be introduced into practice. Furthermore, as Dr. Clayton mentioned, such results are commonly obtained under experimental conditions that do not meet generally accepted standards for diagnostic clinical laboratories. Accordingly, such results, as a rule, should never be considered diagnostic and should never be entered into a medical record or communicated to a tissue source. In the event that research yields information that investigators believe could be of significant consequence to particular individuals, the investigators should bring that matter before the cognizant IRB to determine whether, how, by whom, and to whom results might be communicated. 3, in the public deliberations to date about genetic privacy in human tissue samples, a tremendous amount of effort has been expended in trying to deal with the thorny issues of informed consent. I think this effort has been largely misdirected, and further argue that continuing to wage battles over the bioethical and legal niceties of informed consent is not an effective, practicable way to resolve the central issue of how to give due respect to the protection of patient privacy, while at the same time continuing to facilitate research access to archival patient materials. And I just want to make the point that when you talk about archival patient materials, you're talking about records as well as tissue samples, as I will elaborate in a moment. So it's a bigger issue than tissue samples. This nation has made a substantial and enduring public commitment to the support of biomedical, behavioral, epidemiological, and health services research. And there is a compelling public interest in ensuring reasonable access to the materials required to pursue that research. Current regulations and practices involving informed consent have been elaborated with great care to deal with circumstances involving the direct interaction of investigators with prospective human research subjects in a context in which the stringent requirements of the informed consent protocol are both reasonable and feasible. That is, it is possible to tell the prospective subject in detail the objectives, duration, procedures, risks, and benefits of the study, whether the investigators have conflicting personal, financial, or other interests, and any other matters deemed appropriate by the investigators of the cognizant IRB. None of those circumstances obtains with respect to tissue specimens excised for medical reasons, or for that matter to any archival patient materials that are subsequently used in what is generally referred to as secondary research. With respect to tissue samples, which can be preserved indefinitely and have been accumulating in the archives of some major academic medical centers for over 100 years, it is impossible to predict or even imagine, let alone describe, the kinds of research questions or experimental technologies with which those samples might be involved in future months, years, or decades. Accordingly, for such research, an informed prospective consent of the stringent kind routinely used in direct interaction with research subjects is literally an impossibility. Now, to deal with this problem, 3 alternative generic approaches to the issue have been proposed. The first is embodied in a generally but not universally accepted proposition that different stringencies of informed consent should apply to the use of tissue samples depending on whether they are linkable or identified or linkable to specific individuals. Many believe that a minimum burden of informed consent should apply to samples that are truly anonymous, that is, totally and irreversibly unlinked from their sources. And you could say maximum facilitation of tissue-based research could occur if only the tissue samples could all be routinely anonymized. But this is unrealistic, given first that the major source of those tissues is a national archive that is overwhelmingly comprised of specimens removed for medical reasons and must remain identifiable for future access on behalf of patients, just as the patient's medical record must remain identifiable. Now, although sample anonymity is compatible with some research protocols and with all uses of such tissues for professional education, in many studies, especially in pathogenesis research, it is often necessary, both in developing the protocol and particularly after the results are in hand, to obtain additional or follow-up clinical information in order to establish the diagnostic or prognostic significance of the findings. Indeed, absent the availability of that correlative information, the utility of the primary data may be nothing more than a curiosity. So although the identity of subjects almost never needs to be known to investigators, in fact, essentially never, the research materials, whether, again, they're tissue samples or records, must remain linked to patient sources. So anonymity is not a general solution to this problem. The second generic approach calls for specific reconsent from the patient source or next of kin for each resource use of a tissue sample. Given the age distribution of the collection of specimens, decades, 100 years old, that may be involved in these studies, considering the large population that must often be accessed to amass sufficient experimental material or to ensure that the sample is appropriately structured and unbiased, most believe, not all, but most believe, that the requirement for serial reconsent is of questionable feasibility and would be chillingly burdensome, if not lethal. Now, I know that Dr. Clayton disagrees with that, and I respect her difference, but I'm not sure I can go along with it. The third generic approach would adapt the concept of stringent informed consent by substituting for an informed discussion of a specific research protocol speculation about hypothetical future uses of tissue specimens and inviting prospective subjects to direct, limit, and prohibit the future uses of those specimens for research. Such an approach, if widely implemented, would essentially cripple the future research flexibility and utility of the tissue archive. Moreover, to introduce such discourse into a clinical setting typically freighted with anxiety is to risk transforming a process designed to accomplish informed consent into one that would too often lead to uninformed denial. 4, there is general agreement that the current practice of burying in routine hospital and operative consent forms language permitting the use of medically removed tissue specimens for research and education is inadequate. It is inadequate. It must be strengthened. There is no debate about that. However, research on archival human tissue samples, whether linkable or not, should continue to be governed by an appropriately crafted, general, informed consent procedure. The language should be clear and simple, based on fact and reason, not speculation and emotion, and structured on the premise that the tissue archive has always been and should remain a public resource dedicated to the public good, not a savings bank with deposits of private property. 5, as a necessary corollary to that recommendation, there must be significant strengthening of the protection of genetic information, and indeed of all medical information created in research, from unauthorized disclosure and hurtful misuse. This is essential both to help allay public unease, to sustain public confidence in research, and to ensure that research conducted on archival tissue samples and other patient materials is indeed of minimal risk. One possible mechanism for accomplishing this objective might be modeled on the statutory certificate of confidentiality in conjunction with a new assurance mechanism requiring that all academic and other institutions engaged in human subjects research or in secondary research on archival patient materials have in force an institutional confidentiality policy that had to meet federal standards. Among these would be unambiguous measures for protecting from inappropriate or unauthorized disclosure the confidentiality of medical information created in research, whether genetic or otherwise, the provision of severe penalties for violators, the existence of credible mechanisms to secure identified or identifiable medical information from unauthorized trespass. Of special importance would be the need to establish within each institution secure mechanisms that sequester and protect the security of the locus at which linkages between identifiable codes and specific patients are maintained. Under the assurance mechanism, institutions with approvable confidentiality policies would be awarded a certificate of confidentiality or something similar that would grant protection to the institution and its research staff from the forced disclosure of any confidential medical information created in research. The existence of such a mechanism would provide important assurance to human research subjects that identified or identifiable medical information developed in research could not be forcibly disclosed without their express authorization to anyone, including insurers, employers, family members, healthcare organizations, or government agencies. And 6, the embrace of the certificate of confidentiality in statute is explicitly restricted to research records. It does not protect clinical records used in patient care delivery. This raises an important point because the medical record often contains genetic information, whether derived from the medical or family history, from routine clinical laboratory studies, or from the direct analysis of DNA sequences. Accordingly, discussions of the issues of genetic privacy and securing the confidentiality of genetic information inevitably reaches the medical record and the much, much more difficult issues of the privacy, confidentiality, and accessibility of medical information. And this also means that the population of researchers at risk from proposed new statutes or regulations directed at the security of genetic information extends well beyond pathologists and, quote, genetic researchers to include all those investigators for whom medical records constitute primary research materials and databases. Although there exists a complex web of statutes, regulations, accreditation standards, and professional codes that address the confidentiality of medical information created in the clinical setting, medical records have in fact become routine research commodities, whether for purposes of health services, epidemiological, or clinical research, for public health purposes, or by authorization for purposes of oversight, regulation, and accreditation of the healthcare delivery enterprise. It is veritably impossible today in this country to obtain medical care from a healthcare provider, an individual, or an institution without, on first encounter, having to sign a brief, simple, general release that authorizes widespread access to one's medical record for insurance and a host of other purposes. You cannot get care without signing that release. So for these reasons, I'm convinced that to provide maximum security to medical information developed in research on human subjects and from patient-derived archival materials, we must build a firewall around the research data and ensure that those data do not freely commingle with clinical patient care records. Although such a firewall can be conceived in specific circumstances to have troubling consequences, and even on occasion to raise thorny ethical issues of investigator responsibility, I believe that the alternative, that is, to permit any regular channel between clinical records and research records, would fatally impair the protective net that is the heart of the proposed assurance mechanism and would imperil the confident assurance of confidentiality to potential research subjects. Thus, I conclude that the trade-off would be well worth it if it permitted the continuation of genetic research on human tissue samples, as well as other secondary research on archival patient materials, under a general consent mechanism that would afford maximum flexibility to future research opportunities and needs. Thank you.
Before we move on to formal debate or informal responses and questions from the audience, I've been asked to remind you that those of you who would like CME or CNE credits for participation in the seminar need to fill out the appropriate forms and hand them in at the registration table at the front of the auditorium. In addition, if you would like to submit questions on cards rather than struggling to the microphones, assuming you're in the middle of the rows somewhere, please do so. There's folks who will distribute the cards and collect your questions. Okay?
Actually, let me begin by pointing out the very substantial areas in which Dr. Korn and I agree, because they are many. First of all, I wholeheartedly agree that the distinction between genetic and non-genetic research is essentially an untenable one, and that the reason that the document that is included in your handout addressed genetic research is because that's what we were asked to address. And the concerns, I think, are actually quite similar. I also agree that linkable information is in fact what is almost always required for research, that making samples totally anonymous is not the solution. I also agree that it's critically important to separate medical and research records. And I also agree that there has been way too little emphasis placed on protection of patients. So I think that all those things are areas on which I totally agree with him. Here are the areas in which I disagree. First of all, I think that while patient confidentiality is incredibly important, it's not the only thing. That in fact, there is a difference between patient care and research, and because our laws suggest that informed consent ought to be required, it is important that our practice differs from what the law requires. Now, I will tell you that there are others who are very happy with the way the legal requirements are currently set forth. I at least am willing to entertain the possibility of readdressing these issues and in fact saying to patients when they come in and seek health care that just by the way, whether you like it or not, your records are going to be used for medical research and your tissue samples are going to be used for medical research. But I think at least until we get to the point of changing the laws or put in place a process of actually allowing patients to decide whether they want this to go forward or not, we have to look at these issues. I, by the way, do agree with him that a fairly general consent looking to the future is in fact the way to go. And that in fact is what the National Action Plan has been attempting to develop, and that process has involved extensive input from pathologists and scientific researchers. So I just want to put that on the table. And then, and I would also say that there is no evidence that uninformed denial would prevail if patients were in fact asked to consent at the time they get, give consent for their medical care. So I think that let me just be really clear, I think, about what the areas of debate are between us. One is the notion that our laws appear to require some kind of consent, that is in fact not the current practice and that we need to figure out how to answer that question. And I think the other thing that is really important to acknowledge is that at least at present, a lot of research goes on using medical records, using stored tissue samples without any supervision, without going to any IRBs at all. And at a minimum, I think we at least need to say that there needs to be some sort of supervision of this sort of research, and that we need seriously to consider asking our patients whether that's okay. If that's not a posture that we want to adopt, then we need to change our laws. But that's essentially where I end up at this point.
Yeah, I believe informed consent is necessary for research on archival tissue specimens. I also share Dr. Clayton's concerns, perhaps I even have greater ones, that there's a huge amount of unconsented, if that's a word, examination, if not research, of medical records that goes on all the time in our healthcare system, and it's getting increasingly prevalent, not decreasingly, and that I think there needs to be more discussion and consenting and oversight and authorization of those practices as well. However, back to the tissues, the main difference that I have, I think, with Dr. Clayton is that when I use the term general informed consent, I mean general, and I think when she uses the term informed consent, she has more elaborate provisions in it than I'm comfortable with. I tried a couple of weeks ago, just because I had a spare hour, to write out an informed consent form that I would be happy with. And it basically consisted of a page that explained the whole issue of tissue saving and using tissue for research, and a little bit of sales about the good things that have happened from being able to do research on this tissue and so forth. And then it had a section that gave the patient a very clear choice of agreeing that tissue unneeded for diagnosis, etc., could be retained for purposes of research and education and such. Yes or no. And if a patient says, no, I do not wish my tissue to be used, then we have to respect that. I've never believed that you just do it over a patient's objection or whatever. So I agree on consent. What I don't like is to start getting into these Talmudic discourses about the possible future uses, and no, I don't want my tissue used for this, and I don't want it shared with that, and I don't want this, and I don't want that, about things that you can't even discuss intelligently because you don't know what they're going to be. And that, I think, is an invitation for limitation of a resource that is gratuitous and unnecessary. And I think it's unnecessary if we can protect the confidentiality of the information and keep it from getting linked back to the patient in ways that would be injurious to the patient or his or her next of kin. That's the difference.
Well, Dr. Korn, let me say that we're even closer than you think. The National Action Plan form gives patients 3 choices. First of all, do they agree to research on the disease for which they were hospitalized, or actually cancer more generally? Second, do they agree to research on other diseases? And third, are they willing to be recontacted in the future to participate in research protocol, in specific research protocols that involve getting more tissue, getting more information directly from the patient or not? So those are the choices that exist under the Action Plan for Breast Cancer. Those are the choices that they have. It's not who it gets shared with, whether it's genetic or not, or anything like that. And the reason for thinking that way is that we surmise that most patients who have a serious disease actually are pretty interested in people figuring out how to solve their disease. They may have more trouble with other sorts of diseases, particularly those that carry social stigma, although they may be willing to consent to that as well. So I honestly think that the direction that we are going is, in fact, very close to where you want to go.
Good.
Since for the first time in hearing about this debate, there's been so much agreement between the two, I want to focus on an item that may result in a little bit more agreement, perhaps. In both presentations, there was it seemed to slide over the issue of linkage with where the linkage was. Specifically, I did not hear that anyone wanted to make the existing medical data or samples no longer linkable, that is to say, anonymous and without identifiers. The regulations, however, say that what the researcher receives is supposed to be anonymous. It is possible, and in fact in the Indian Health Service we have done it. We have supplied linked data, specimens plus medical information linked to that specimen in a way that the researcher, it was totally anonymous to the researcher then who that person was, as well as to the institution that gave it. This was actually in Anchorage, both the CDC laboratory that had the specimens and the Indian Health Service hospital that had the medical data. So that no one knew what the given specimen was that the researcher received. There was no way that anyone could know, either the researcher or the institution or the CDC lab, who those specimens were. It seems to me that process complies with the regulations and permits the kind of research that Dr. Korn wants and provides the protections whether or not we agree with the regulations or not that the regulations have. And I'd like a comment from both of you.
Dr. Freeman, are you suggesting that once the information is supplied, that there is no way, that essentially you're talking about making, anonymizing samples, to use the unfortunate word that we used in our...
Anonymizing the linked data, the specimen with the data, in a way that now the researcher receiving it cannot know that it came from me and will not be able to contact me. And there is, no matter who that researcher asks at the institution or the pathologist, they don't know either.
But someone in the universe knows?
No one knows.
Okay. I mean, I would say that that complies with the regulations. The only, I mean, I'd say that you're right about that. I think that it is important to recognize that it is an ethical step to take samples that are currently identifiable and make them anonymous. But I think as a matter of law, as a matter of law, that once they are really made anonymous, then I think that it's probably the case that they, that neither IRB review nor patient consent is required. I have to tell you that one of the things that I'm obviously strongly interested in patient consent. I am actually perhaps even more interested in sort of the amazing kinds of research that I see people doing with medical records and tissue samples without asking anybody anything. I heard that in the Northeast, when you, at least in one major medical center, when you go, you have a patient with disease X, and you go and do your literature search, not only do you get the peer-reviewed literature search about disease X, but you also get the medical records of the last 3 patients in that institution who have disease X. Now, first of all, talk about useless information. That strikes me as really useless information. But second of all, it is a breathtaking breach of patient confidentiality. I mean, so I think and the ability to do research and just kind of go in and pull up an ICD-9 code, and everybody who's had that disease in your institution in the last 15 years is just stunning. And and so truthfully, the concerns that I think that Dr. Korn and I would actually, I know that we would both agree on, are that those are huge problems that need to be addressed, and that we really do need some sort of oversight and some sort of recognition that this isn't the way it's supposed to be. I mean, I'm very confident that you would agree about that, because I'm amazed at the stuff that people pull up out of medical records without asking anybody anything, even though it's required by, not only is it legally required, but it's just ethically required. It's not the case that when you go in to get health care that you agree to become a research subject automatically.
Question. In that Northeast institution, did you determine whether or not the 3 cases that were listed in the database had given consent for that information to be listed in the database before you came to your conclusion?
I can't imagine that they were.
I don't know, but if they had given consent for their information to be put in the database, would you be reconciled?
The truth is, insofar as violating their rights are concerned, I suppose I would be reconciled. As a way to direct future patient care, I think it's just ridiculous. Suppose the last 3 cases were grossly mistreated. It's just not good data. We are trying to move toward evidence-based data with peer-reviewed, rigorous scientific methodology, and that ain't the way to do it. So I just think it's I think it's crazy, but I would be more reconciled to it from the patient's perspective if they had agreed, but I can't fathom that anybody would agree to that.
We have a variety of topics submitted on cards, so perhaps I could get to a couple of them. Here are 2 that address similar issues. Neither speaker touched on the issue of commercialization of information or products derived from tissue versus the issue of consent. And another person asks, how does the use of informed consent to protect individuals and groups against undesired, in quotes, vacancy provisions of patent laws when genetic material is collected?
The issue about commercialization is obviously an important one, and is one that I chose not to talk about. I will say, but let me say this. I think that the issue of talking about property rights is really misleading. I think that the issue, at least with regard to research, is one of stewardship and who ought to have some control in it. I think that probably the Moore Court was right in saying that it would be a bad policy to say that just because a tissue sample is obtained from you, you get a right, commercial right, you know, into perpetuity in the future in the developments of that tissue. I think that, you know, that there are things that scientists do, very important things, that turn that into a commercially viable product. My own view about this is that people need to be told that that things get developed and they make people money, and my own posture that I've taken is, and they don't get part of it. So, I mean, there's obviously a huge debate about that, but but whatever we think about the patient's interest in the particular tissue, particularly when there's been dramatic transformation of that tissue to develop new data or something of that, you know, I think that their commercial interest in it goes away.
I agree with that, I have to say. But on the other hand, I have a problem with mass commercialization of institutional archives. And to be very specific about it, since it's a matter of public record, when I read in the New York Times a summer or two ago, I think last summer, that New York's Memorial Sloan Kettering had entered a joint venture with Sequana, which is a genomics company around La Jolla, to form a 50-50 for-profit enterprise in which Sequana was going to bring to the table its expertise and technology in doing genomics work, and Sloan Kettering was bringing to the table their tissue archive, which was the asset that was buying them 50% of this for-profit venture. I got real sick of, I mean, my stomach turned on that one. I don't think that's right. I wished it had been a not-for-profit deal. I would have felt a little better about that. I think that what you have to realize is that the country, for better or worse, has decided that promoting translation of research property into commercially useful goods is a major public benefit, and therefore all kinds of incentives are out there to push that traffic. And there's really not much we can do to prevent it at this time, except to try to modulate it so that it doesn't transform academia into just another crass industry, which I worry about. But I don't think that people should have rights to the goods that come from way downstream work that might lead to the development of a product.
All right, another question was asked, have you any examples of individuals who were damaged by retrospective, i.e. archival research activities as they are currently practiced? In other words, is this a theoretical debate about potential harms rather than an actual historical debate based on real harms?
Well, let me answer that in two ways and then turn it over. I have colleagues who have operated NIH-sponsored tissue banks, which are extramural tissue repositories for research, for years and years and years, who claim that there has never been a breach of security or any kind of adverse impact on a patient whose tissue wound up in the repository. On the other hand, there are some statements and some papers out of anecdotes, always anecdotes, but that doesn't mean they're not important, of stupid behavior. It's just like misconduct, you know? I mean, people do really stupid things. There's a classic story of a researcher who called somebody out of the blue that he didn't know and they didn't know him and said, hey, gee, you got Huntington's disease in your genome or something. And that's just plain criminal and stupid. So there are these anecdotes out there of people whose common sense is lacking, and that's what feeds a little bit of this.
I will agree with that. I think that the main issue is people calling patients up with specific identifiable research results. These researchers often feel like they're doing the right thing, you know, to say, well, you know, this is really important information and they really need it. And I think we have to deal with that. And I have to tell you that researchers I know feel incredibly uncomfortable when you say to them, you know, you just can't go calling folks up and tell them stuff that they didn't ask you to get. And I have to say that Dr. Korn has very appropriately said in the article about use of stored tissue samples that occurred last year, I mean, this is just bad practice. I realize that it's hard to have to deal with just stupid stuff, but it is, looking at it in the healthcare system in 1997, it's just stupid to do that. So yes, there are real harms.
I've been most lacerated on that position by some very distinguished academic pediatricians who feel that they're bringing God's word to the patient if they can tell them about some research result, and they feel very passionately about it.
Everyone who uses genetic testing in quotes by clinicians, in quote, has found that the clinician using genetic testing is completely stupid about it. 60% of people who order genetic tests and telling patients what they need do not understand what we're talking about. And those are supposed to be people who care about patients. And we know researchers.
Right.
They don't.
Some of them do.
Major Vanderham.
I'd like to ask a couple military applications. One, I thank you for your background on 10 U.S.C. 980, and I give one personal interpretation, one military person's interpretation. This isn't the whole department or any of the services. The words that are used in 10 U.S.C. 980 talk about a human being being used as an experimental subject. In my own interpretation, when we're using existing data in a record or a tissue sample, that is not a human being being used as an experimental subject. However, they are research subjects under the Common Federal Rule definition. But I think in my own interpretation at times, we can say that 10 U.S.C. 980 does not necessarily apply for tissue research or data research. But I want to get away from that. That's just a statement. The military application I want to ask is, we in the military have to come in and give our DNA samples. We have to give our blood samples for HIV. There's certain things we have to do that are not necessarily our free choice. Do those serum repositories or those databases that contain our specimens, do they need the same protection as regular pathology specimens in a hospital, or do they need extra protections in a military setting where we didn't freely give those samples?
I think the answer to your question is probably yes, that they probably do need special protections. And I partly say that looking at the, at the cases that have come out, particularly from the DNA repository, where it was very important to the court that the reason to get these samples was for identification of personnel who are killed in that line of duty. And it was very important to the court that these samples were not going to be used for research, and that they distinguished away 10 U.S.C. 980 as well as the federal regulations, saying that this was not what was at stake here. So I would say that it's also been important that, as I understand it, the military has taken the position quite clearly that the only thing that the DNA repository is going to be used for is this posthumous identification. And I think that, to be honest, it's hard to step into the mind of judges, but it would seem to me to be it would seem to me to fly in the face of that particular case to then say, well, we've got these samples and we got them involuntarily, and we can use them for research without getting informed consent. I think the issues of consent in the military are really vexing because you live in a culture where voluntariness is not, and individual autonomy does not hold the same valence that it does in the culture at large. And the idea of pitching in and being a good soldier is a very important ethos in the military. So I think that the issues of voluntariness are complicated at best, but particularly in a setting like this where the specific purpose of the repository is for a specific military purpose, you would be, I think, hard-pressed to say that these samples ought to be usable without going back to the individual service personnel and getting their consent.
Let me ask a question, just because I think it's a very interesting problem. What would happen, or what would your reaction be, Ellen, if a portion of each sample were truly anonymized with a minimum amount of descriptive information, maybe the age or gender, whatever, may not even need that, of course, and used as an anonymous sample of, as an anonymous collection of samples for whatever future research uses might be appropriate? Would that be appropriate? Anonymous now, really anonymous. You can never connect the sample back to the person who volunteered it.
I think it would be legally permissible. I think it raises some real serious questions, but I mean, I think the law would permit it.
What would the major think of that, who asked the question? Where is he?
Personally, I have some problem with it. Some of my question relates not only to the DNA repository, which we've had a lot of interest in, but also we have an HIV serum repository, and we've had a lot of universities and other organizations very interested in getting access to these active duty military specimens that were collected on order. And we have whole tracking for years and years and years, and it's an excellent database.
That's right. That's why I'm asking the question.
Let me also add that these repositories are going to grow as we develop pre-deployment, deployment surveillance to avoid some of the problems associated with past military experiences. There's going to be huge repositories of serum and materials. Yeah.
There's a forensic concern, ethical concern, in retaining samples in the repository. If those samples are retained for long periods of time after the soldier is separated from military duty, and crime scene evidence is compared with the samples of only the military repository, since there isn't a similar civilian repository, the chance occurrence would be very low of identical gene riplets looking the same, imputing a military person as being a criminal when in fact his civilian counterpart has gone free, could occur, theoretically could occur. I feel that that the retention of DNA samples from military personnel should separate from the service at the same time as the military person when he either retires or leaves active duty permanently, and should not be retained after he separates from the service. I don't know if that policy is approved.
One last question.
Just so our esteemed guests can see, there's probably as much difference of opinion among the military as there are amongst other academic scientists and epidemiologists. If the issue is, if they bring in protocols that are good science, and if they're truly anonymized samples, then I think there are instances, and I would have no problem using that kind of data, and see no difference between using the data you have in your tissue banks in the civilian world on proper protocols with appropriate confidentiality and taking the steps to make it truly anonymous.
Just one follow-up on the DNA or the serum or the tissue repository, as we call it now, to get it away from the DNA. The Assistant Secretary of Defense for Health Affairs has a very stringent policy on access to that. I think that was in relation to the court case in Hawaii, where access to that is limited solely for remains identification, that there are very stringent requirements on even relatives requesting it be used because there's even issues of whose parents whose, that controls access to that, unless there's a duly instituted court order, and it still has to be approved by the Assistant Secretary of Defense for Health Affairs. So those are very stringent. It was collected for a specific purpose. Now, when we get to some of the repositories like serum or tissue, the purposes for which they were collected aren't quite as clear. But the DNA, also, Art, you can, when you leave your commitment to DOD, which might not be active duty, but even retirement, you can request to have that removed from the repository. And even the backup specimens, which used to be in the health records, are now being removed.
All right, well, it's past 10 o'clock. According to my schedule, we're into the coffee break. Please reassemble at 10:30. Thank you.